Hello and welcome again to week 1 physiology. Next thing that we're going to look at, we're going to kind of combine two things just so that we can keep the last video a little bit shorter. We're going to talk about membrane potential and the sodium-potassium pump. So let me find specifically what we want. So we mentioned membrane potential already when we talked a little bit about that Nernst equation, how it's that electrical potential. It kind of looks at the difference between the electrical charge, the overall charge, inside compared to the outside. We also mentioned very, very briefly, let me find here. We mentioned very briefly that the neurons, we typically say that those are around -70. That's the value that we're going to stick with, -70 for neurons. Smooth fibers, we typically use -60. Skeletal fibers, we usually use around -90. And I believe cardiac cells as well, cardiac muscle cells, are also around that -90 mark. These will be important later on. We'll jump back into these. The key one that we want to look at now is that kind of -70. That's the one we're going to focus on with this lesson. Getting a little bit ahead of us, though, ahead of what we were talking about. So we have what's called that membrane potential, right? Membrane potential is that difference in electricity. We can measure that. We talk about how it's positive on the outside, more negative on the inside because that negative from those proteins. Jumping into a couple of definitions. Where the cell typically sits, where it maintains, where it finds balance at rest, is known as the resting membrane potential. So when nothing is happening to the cell, it's going to sit at resting membrane potential. Remember, for neurons, that's at -70 millivolts. This is really where, I guess, just kind of showing this again, all of these different factors, those chemical forces, those electrical forces that we described in the previous videos, as well as, some of that permeability stuff, that Nernst equation, that Goldman equation, all of these guys contribute to membrane potential. Now, just like we talked about, the main ions that we look at is that there's a lot of sodium outside, and there's a lot of potassium on the inside. In the next video, we're going to look at action potentials, and we'll see that with action potentials, with messages that get transferred, the way that happens is that we let a bunch of sodium into the cell, and we let a bunch of potassium out of the cell. So we need a way to kind of reverse that, as well as maintain our membrane potential with some of the other things going on, like leaky channels and those kind of things that we mentioned as well. That is where our sodium-potassium pump comes into play. The sodium-potassium pump, you can also see it written as sodium-potassium pump, or the sodium-potassium ATPase, anything like that. This is how you can see it written in any of those. And remember from Biochem 1, ACEs are enzymes. ATPase specifically is the enzyme that's going to break down ATP, that adenosine triphosphate, which is our main source of energy. So pause, back up. If we are using a protein and we are using energy, what kind of transport is this? Because we're using energy, it is going to be active transport, and because we are using a protein, it is facilitated. So this is active facilitated transport that we're going to be looking at. So with that active transport protein, what we're going to do is we're going to pump 3 sodium out of the cell and 2 potassium into the cell. And remember, when we talk about active transport, we use energy and we go against the concentration gradient. And that is the main thing. It's not super complicated, but those are the key points that I want you to remember with this, with the sodium-potassium pump. This is really, so the function, these are the three functions. It establishes that resting membrane potential. I promise you this is a question. The purpose of the ATP, of the sodium-potassium ATPase, is it establishes the resting membrane potential. It also is going to help control the cell volume, like we're talking about that hypotonic solution. It made sure it helps to maintain that balance. And then this last one, the new professor doesn't really talk about this at all, so I'm not going to get into that very much right now. So really short. This one video is pretty short. That's really all. Just remember 3 sodium out, 2 potassium in, 3 sodium out, 2 potassium in. Remember that. That's going to be kind of a test question. And yeah. So with that, we'll go ahead and end this video really quick. The next one we're going to start looking at is action potentials. Action potentials are going to take a little bit longer to wrap our brains around, but it shouldn't be too bad.